The root is one of the key organs that helps distinguish between a dicot and a monocot plant. Let's observe the transverse section of the matured zone of roots to understand their internal structure. We'll begin by studying the anatomy of a dicot root. The outermost single-layered tissue of the dicot root is called the epidermis. On the epidermis, you will also find some extended epidermal cells, which are unicellular root hairs. The epidermis is followed by cortex, which is formed of several layers of thin-walled parenchymatous cells. These cells have intercellular spaces. The innermost layer of the cortex comprises barrel-shaped cells without any intercellular spaces. This layer is known as the endodermis. The tangential and radial walls of the endodermal cells have deposits of a water-impermeable material or suberin, which is also called Casparian strips. Suberin is a waxy, waterproof substance present in the cell walls of the cork tissue in plants. Going back to the transverse section of the dicot root, after the endodermis, a few layers of thick-walled parenchymatous cells are present in the tissue. This is known as the pericycle. Secondary growth, such as initiation of lateral roots and vascular cambium, takes place in the cells of the pericycle. Notice that the pith in the tissue of the dicot root is extremely small and undeveloped. Typically, there are 2 to 4 xylem and phloem patches in a dicot root. In the later stages, a cambium ring gets formed between the xylem and the phloem. Some radially arranged parenchymatous cells are found between the vascular bundles. These cells form the conjunctive tissue, which is specialized for storage of water. The tissues present inside the endodermis, namely the pericycle, vascular bundles, and pith, are collectively known as stele. Let's now take a look at how the monocot root is organized internally. In many aspects, it is similar to the dicot root. On comparing the two roots, we find the monocot root also consists of the epidermis, cortex, endodermis, pericycle, vascular bundle, and pith. However, there are some differences. The monocot root can have many xylem bundles, whereas the dicot root does not have quite as many. Moreover, in a monocot root, the pith is well developed. Although dicot and monocot roots have some structural similarities, they differ in the number of xylem bundles and size of the pith and cortex. Moreover, secondary growth takes place only in the dicot root. In other words, these roots thicken over time. A stem is the main axis or stalk of a plant, and it develops from the plumule of a germinating seed. Let's examine the internal structure of a stem in dicot and monocot plants. Here is the transverse section of a dicot stem. The regions of tissues in this stem include epidermis, cortex, and stele. The outermost layer of the stem, or epidermis, consists of a layer of closely packed cells and is covered with a thin, waterproof layer called the cuticle. The epidermis also contains stomata and hair-like outgrowths known as trichomes. The next region of a dicot stem is the cortex. It lies between the epidermis and the pericycle and consists of multiple layers of cells. The main function of this region is storage of food material. The cortex comprises three subzones, namely the hypodermis, the cortical layers, and the endodermis. The hypodermis is the outermost zone of the cortex, which consists of a few layers of collenchymatous cells. These cells strengthen the young stem of a plant. Below the hypodermis are cortical layers of round and thin-walled parenchymatous cells with intercellular spaces. The third or the innermost zone of the cortex is known as the endodermis. It is a single layer of tightly packed rectangular cells which are rich in starch grains. Therefore, this layer is also known as the starch sheath. Let's now examine the composition of the stele, which forms the last region of a dicot stem. This region consists of the pericycle, the vascular bundles, and the pith. The pericycle is composed of sclerenchymatous cells, and it lies on the inner side of the endodermis above the phloem cells. These cells act as a strengthening material. The vascular bundles are located in a ring on the inside of the pericycle. This ring arrangement is a distinctive feature of the dicot stem. There are layers of radially arranged parenchymatous cells in between the vascular bundles, which constitute the medullary ray. Each vascular bundle in a dicot stem is conjoint, open, and consists of endarc protoxylem. The central region of the stem is filled with pith. It is composed of a large number of rounded parenchymatous cells with many intercellular spaces. Now that you've learnt about the dicot stem, let's study the monocot stem. The tissues of dicot and monocot stems are quite similar. However, there are some differences as well. In the monocot stem, the hypodermis is composed of sclerenchymatous cells, while in a dicot stem, it is composed of collenchymatous cells. The monocot stem has scattered vascular bundles, and their arrangement is conjoint and closed. Moreover, water-containing cavities are present within the vascular bundles. On the other hand, in a dicot stem, the vascular bundles are arranged in a ring as conjoint and open. In a monocot stem, each vascular bundle is surrounded by a sclerenchymatous bundle sheath and a parenchymatous ground tissue. Moreover, the vascular bundles on the periphery are smaller compared to the vascular bundles in the center. Therefore, the essential differences between a monocot and dicot stem lies in the arrangement of the epidermis, ground tissue, and vascular tissue. Leaves are the most visible organs of a plant. They perform many crucial functions such as photosynthesis and transpiration, in which their internal structure plays an important role. A dicot leaf is also known as dorsiventral, as its upper and lower sides are different in structure. On observing the transverse section of a dicot leaf, we find that it has three main parts: epidermis, mesophyll, and the vascular system. The epidermis covers the upper and lower surfaces of the leaf and is coated with the cuticle. The epidermis covering the upper surface of the leaf is called the adaxial epidermis, while that covering the lower surface is called the abaxial epidermis. Typically, the abaxial epidermis has more stomata compared to the adaxial epidermis, which sometimes may not have any stomata at all. The next part of the leaf is the mesophyll, which is located between the upper and the lower epidermis. It is also called the green tissue of the leaf, as it contains chloroplasts. The mesophyll is made up of parenchyma and performs photosynthesis. In dicot leaves, the mesophyll tissue is differentiated as palisade parenchyma and spongy parenchyma. The palisade parenchyma, located near the adaxial epidermis, is made up of elongated cells arranged vertically and parallel to each other. These cells have fewer intercellular spaces. The spongy parenchyma is located just below the palisade parenchyma, and it extends up to the abaxial epidermis. It consists of round cells which are loosely arranged and have large intercellular spaces and air cavities. The third part of a dicot leaf is the vascular system comprising vascular bundles. When observed under a microscope, vascular bundles can be seen in the veins and the midrib of the leaf. The size of the vascular bundles depends on the size of the vein. In dicot plants, the leaves have reticulate venation, in which the veins are of different sizes. Therefore, the vascular bundles are found irregularly scattered in the mesophyll. In the large veins, vascular bundles are usually surrounded by a bundle sheath. Let's take a look at a monocot leaf now. It is also known as an isobilateral leaf, which means it is identical on both sides of its axis and shows parallel venation. The anatomy of a monocot leaf is similar to that of a dicot leaf. However, there are also some key differences. Unlike a dicot leaf, where stomata are found in greater numbers in the abaxial epidermis, in a monocot leaf, stomata are present on both surfaces of the epidermis. Moreover, the mesophyll cells are not differentiated as palisade and spongy parenchyma in a monocot leaf. The vascular bundles in a dicot leaf are irregularly scattered due to reticulate venation. In a monocot leaf, however, they are arranged in a parallel manner in the mesophyll due to parallel venation. In the leaves of monocots such as grasses, veins and cells of the upper epidermis modify themselves as large, bubble-shaped, colorless cells, also known as bulliform cells. On absorption of water, bulliform cells become turgid and expose the leaf's surface. Conversely, in water-stress situations, there is loss of turgor pressure and bulliform cells become flaccid. As a result, the leaves curl inwards to minimize water loss. Hence, a lot of activity goes on in the internal structure of a leaf. Since major food production takes place in leaves, they are aptly known as the plant's powerhouse of energy.